Polypeptide synthesis is the combination of amino acids to form proteins, which have extremely broad purposes and functions that are essential for growth, repair, structure, function, and regulation of the body. Amino acid chains have varying lengths. Peptides are chains of 2 to 50 amino acids, while polypeptides are chains of 50 plus amino acids. Proteins are the biggest of the three, and they are 3D in structure. There are 3 steps in polypeptide synthesis: transcription, translation, and protein formation. Transcription involves the production of mRNA to be used in translation. Translation is the production of proteins for the instructions of mRNA. Protein formation involves the folding of a synthesized polypeptide chain into a specific 3-dimensional structure. DNA is too big to fit through the pores of the nucleus, and its form does not allow for translation. Translation creates a portable form of genetic information that can be used as instructions to create proteins. Nucleotides contain a nitrogenous base, deoxyribose sugar, and phosphate group. These nitrogenous bases form hydrogen bonds with each other. Only adenine can bind with thymine and guanine to cytosine. The helicase initially unwinds the DNA, then RNA polymerase attaches to DNA sequence called the promoter and continues to unwind the DNA. RNA polymerase reads the DNA template strand and builds a complementary mRNA strand. In this newly made RNA strand, uracil, another nucleotide, replaces thymine as the base that pairs with adenine. When RNA polymerase identifies the terminus sequence, it stops creating mRNA. DNA is still rewinding. This forms pre-mRNA. A form of mRNA is not matured and needs to be matured in order to leave the nucleus. Introns, which are non-coding regions, are removed from the pre-mRNA by the spliceosome and a protein and RNA complex. Exons, which are the coding regions, are joined together by the spliceosome. A 5' cap is added to the end of the pre-mRNA, aiding in protecting it from degradation. A poly-A tail, a string of adenine nucleotides, is added to the end of the pre-mRNA. This tail enhances the mRNA's stability. After this process of maturation, mRNA leads through the pores of the nucleus. The events of initiation, elongation, and termination act as triggers that attract the proteins required for export out of the nucleus. The next step is translation. In this process, the mRNA is read in codons, groups of 3 bases, each coding for a specific amino acid. These amino acids are linked in order to form a protein. AUG is considered a start codon, as it is typically your first amino acid in proteins. The ribosome consists of 1 small and 1 large subunit. The mRNA passes through the ribosome where the codon sequence is read and a corresponding tRNA molecule brings the corresponding amino acid. tRNA carries specific amino acids to the ribosome and matches its anti-codon regions on the mRNA, helping build the protein chains during translation. It forms a peptide bond with the amino acid next to it, slowly creating a protein. The first step of translation is initiation. The small ribosomal subunit identifies the initiated tRNA methnion and binds to it. This structure then attaches to the capped structure of the mRNA strand. The small subunit structure and scans along the mRNA strand to identify the start codon, AUG. This process is guided by initiation factors, which detach when AUG identified and the large subunit associates with the smaller subunit. The ribosome has 3 sites during translation: amino acyl site, entry site for the new tRNA carrying an amino acid, peptidyl site holds the tRNA with the growing polypeptide chain, and exit site where the tRNA exits the ribosome after delivering its amino acid. The anticodon region on the tRNA carrying an amino acid in the A site matches against the mRNA code. tRNA with incorrect anticodons are rejected and replaced until the correct anticodon enters the site. The amino acid forms a peptide bond. The tRNA in the P site releases the amino acid onto the tRNA in the A site and becomes empty. Simultaneously, the ribosome moves 1 strip forward. Empty tRNA enters the E site and leaves the ribosome. The peptidyl tRNA is in the P site. The A site is now unoccupied, ready to receive a new tRNA. When a stop codon enters the A site, no tRNA can fit in the A site as there is none that match. The codons are recognized by a protein, a release factor, catalyzing the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome dissociates into subunits. Addition of chemical groups and folding are the steps needed in order to make a protein functional. The shape of proteins determine their function. There are different ways of folding that can happen at different structural levels: primary structure, secondary structure, tertiary structure, quaternary structure. Denaturation occurs when the proteins are not in an ideal environment.